So today we'll give a brief introduction to tangles applied to modeling the action of proteins on circular DNA, but we'll focus mostly on applying persistent homology to viral evolution. Recall that recombinases are proteins that cannot or link DNA because they can cut the DNA in. So we can cut the DNA and thus can change the DNA topology. If these sites are oriented in direct repeats, so head to tail, then when we cut and rejoin, we will change the number of components and go from a knot to a link. But if they are oriented in terms of inverted repeats, where it's head to head, then when this tail connects to this head and this tail connects to this head, we'll have inverted the DNA sequence. And so we can take a look at the DNA sequence with the new black head over here, the white head over here. If I put arrows on this, the DNA, if I orient the DNA like this, well, white to black, I've changed the orientation of one DNA segment with respect to the other DNA segments. So I've inverted that orientation, and so that's why it's called inverted repeats. There are many different types of recombinases. For example, Cre is a type of recombinase that can act on both inverted and directly repeated sites, so it can create both links and knots. Depending upon the reaction conditions, it might mostly create unknots and unlinks, but it can also, again, depending upon the reaction conditions, trap an arbitrary number of supercoils here, trapping them, creating knots and links with higher crossing numbers. So it can create a variety of different products depending upon the reaction conditions. XER, on the other hand, produces a unique product when acting on unknotted DNA. It will always trap exactly 3 supercoils right here, and then introduce a fourth crossing, creating this 4-crossing link, which is often called a 4-cat, for a 4-catenane, 4-crossing catenated product. The question, therefore, is how does XER produce this unique product? Randomly trapping crossings, that we can see happening, but creating a specific product, it has to have a unique mechanism to do that. And it will only do deletions. It will not do inversions when acting on unknotted DNA. Well, the way it does it is it has accessory proteins that set up these exact 3 crossings. So we've got helper proteins that will bind on here that will then allow the XER to act on these sites right here, creating this extra crossing. By trapping exactly 3 sites, it determines the orientation right here. Let me clear the screen. With it trapping those 3 sites, you'll note that if I put an orientation on the DNA, so we'll orient here, travel along, travel along, travel along, and so we can see that these 2 sites are oriented in a parallel fashion. So Cre, the site is anti-parallel. In this picture, the site is parallel. Now that is projection dependent. This really should have a 3-dimensional model instead of this flattened 2D image. But in this flattened 2D image, let's assume parallel. If it were to, so in this case, my sites are directly repeated if they are in a parallel fashion. If I had instead put the sites, if I instead reversed the orientation of the site and did inverted repeats, well, then they would no longer be in parallel fashion, the protein couldn't act, and thus we do not get a product. And so that's how it can perform only deletions, is because it sets up a particular geometry. So it uses this topology to set up a particular geometry here, and so it requires the sites to be oriented in a particular way, so that these sites are oriented in a nice parallel fashion in this particular projection. In reality, maybe the sites, we might change the topology, we might change the geometry while preserving the topology, and so we could, for example, add in, I can draw the 3 crossings again, but now maybe I'll add in a twist down here. And so maybe in reality it does act on anti-parallel sites, but topologically speaking, if we're trapping this thing right here, then topologically I can unwind this crossing right here to get to this. And in reality, we should be looking at 3-dimensional conformations, not these 2-dimensional projections, in which case parallel versus anti-parallel is harder to see, but we would get a specific geometry that this trapping these 3 crossings here would give us a particular geometry here, which would be correct for acting on direct repeats, but incorrect for acting on inverted repeats. And thus we've got this topological filter trapping these 3 crossings, give us a topological filter that prevents inversions and only allows deletions when acting on unknotted DNA. Any questions about that? So recall I talked about XER acting on psi sites, so that's the name of the DNA sequences that it's doing the recombination, that it's cutting the DNA is at these psi sites. And it uses particular accessory proteins, helper proteins, to do the reaction. Well, I mentioned that there are many different types of recombinases. Well, some recombinases can also have different actions. So XER, but acting at diff, so if my XER instead acts on diff sites instead of psi sites, and if it instead has different accessory proteins, then it will do a completely different reaction. So XER acting on diff with different accessory proteins, we don't have those 3 crossings trapped. Instead, it basically will act on something like this, and then add this crossing in a different projection. Maybe it actually bound to a crossing and removed that crossing. But basically, XER acting on diff sites will act on a trefoil knot and create this 2-crossing link, then go from this 2-crossing link to the unknot. In each case, where basically you can see that we remove this crossing, so this crossing disappears. When we add this crossing, we can smooth it out, removing 1 crossing each time, going from 3-crossing knot to 2-crossing hop link, 2-crossing link to this 1-crossing knot. The 1-crossing knot simplifies to the unknot, and then we can go from the unknot to the unlink, so to the unlink right here. And so XER will unlink these replication catenanes in this stepwise manner, where it's the same XER that we talked about on this page, but with different accessory proteins binding to a different sequence. How was this method determined? Well, this method, so how were these complexes determined? You know, that it binds 3 crossings, that it did this stepwise unlinking. Well, it used tangle analysis. So let's do a very brief introduction to tangle analysis. when one does mathematical models, usually the real-life situation is too complicated, so we must simplify things. Proteins are really linear chains of amino acids. So a protein, if I were to draw a type of protein, just be a linear chain that folds up on itself into some type of conformation. And so that's what this thing here shows is a protein. Well, in our very simplified model, our protein becomes a three-dimensional ball. In other words, we're not going to care about the shape of the protein for this mathematical model. We basically will just use the protein as a divider between the stuff that's bound by the protein within the ball and the stuff that's not bound by the protein. Our DNA, we will ignore the nice double helical nature, we'll ignore the sequence, and we will just think of DNA as rubber tubing or just an arc. Well, why does this model make sense? Well, here's a picture of protein bound to DNA. And sure enough, the protein looks like a three-dimensional ball right here. And the DNA looks like just, arcs coming out here. And so we can model our protein DNA complex with this three-dimensional ball right here. And the DNA, we've got some segments coming outside here. But then the question that we are interested in is, how do we draw the DNA inside the protein? So this DNA is bound by here. Most likely it wraps around the outside of the ball, but for mathematical convenience we push it inside of the ball so we can draw some kind of shape. And the question is, how many crossings does this protein trap? How many DNA crossings does it trap? What is the shape of the DNA bound by the protein? Well, we can determine that by solving tangle equations. So this is an example of a tangle equation that came from the biology paper Path of the DNA within the mu transpososome. So they modeled the protein as this ball right here, and they wanted to know, how do I draw the DNA bound within this protein ball? Again, it likely wraps around the outside of the ball, but if we can first determine how to draw the DNA inside the ball, we can then push the DNA to the boundary. Once we know the topology, we can apply DNA energetics to get a more realistic three-dimensional model. But first, we need to know the topology. So the thing is, we want to find the topology of the DNA bound by protein. And the actual solution is shown right here in B, that mu traps 5 DNA crossings, and those 5 DNA crossings are in this conformation right here. And you can see that is a solution to this one equation. If I take this thing and plug it in right here, plugging it in, I can see that this crossing here will simplify. I can remove that crossing. It's topologically not needed once I remove the blue protein. And so my product is the 4-crossing link. It's the 4-crossing link that I am expecting. Versus we know that this is not a solution, because if I plug it in, I actually only get 2 crossings left. I don't get this 4-crossing product right here. In fact, this one equation, so in this paper, they gave us many equations with this one unknown of trying to solve for the shape of the DNA bound by the protein. But note that this one equation right here already gives us useful information. If you think about it, what's the minimum number of crossings that T must contain? T has to contain at least 4 crossings, so for this equation, there's no crossings in this outside. The DNA doesn't cross itself. So in order to get this 4-crossing product, you have to have at least 4 crossings inside of here. And so we already know that our solution is going to be biologically interesting, that it'll have to trap at least 4 crossings. And in actuality, it does trap 5 crossings. You can see this paper for a description of the other equations and how they get this 5-crossing product result. So that's tangle analysis. We will now discuss the topology of viral evolution, this PNAS paper published this year, 2013. In order to discuss the paper by Chan, Gunner, and Carson on the topology of viral evolution, we will first need some background. We'll start with discussing some examples of mutations. In this particular example, we have a point mutation. This A gets changed to a C. So a point mutation is where one base gets changed to a different base. Remember that DNA gets transcribed into RNA, which then gets translated into protein. So if I look, for example, at this CAT, the DNA, will get translated into RNA. C pairs with G, A pairs with U in RNA, and T pairs with A. And then this triple right here gets translated into one amino acid to make protein. So one amino acid, and this particular amino acid is histidine. So it will then get translated into the amino acid histidine. Now, there is some redundancy in terms of this notation. So when you do a point mutation, it may or may not affect the amino acid that results. In this particular case, however, changing this A right here to a C changes the amino acid from histidine to proline. And so that could have anywhere from a no to a major effect on the protein. So it could be a minor effect or a major effect depending upon the substitution in the amino acid. But that's one kind of mutation that can happen. One can also have a deletion. Note, a deletion causes a more significant change if it is just a point deletion. So if we take this A and we delete it, now when you're reading the protein, we had that this CAT gave us histidine, CAT, but now we have the A is no longer there, so now it's CTC. So we have this, but now our triple is CTC. Our reading frame has changed. So now we're reading different things, and we're creating leucines, isoleucines, etc. And that can be a much bigger change in terms of your protein. If you do 3 deletions, well then all you've done is deleted. So if we had gotten rid of both the the CAT, then we would just remove one histidine, so that might not have been such a big change. But it can again affect the protein. Similarly, if you have a 1 base insertion, so if we create this A between here, so if we do an insertion right here, again, we're now changing our reading frame. Instead of reading cat, we're now reading ACA, TCA, and so we now get a different amino acid sequence again. And so that can again have a major change on the protein, which could potentially kill the organism, or maybe the change is good and it's positively selected. Most mutations aren't positive, but a few are. Another type of mutation that you can have, which is much larger, is a recombination. So we could have that our DNA sequence changes because we invert this sequence with respect to the other one. Now, we talked earlier today about site-specific recombinases, where you had to have a particular sequence to do the recombination. But there are other types of recombination called homologous recombination, where all you need is for the sequence to be similar. So we just need to have a similar sequence for homologous recombination. So here are two chromosomes. So this red chromosome is actually just one linear piece, that's all nicely packed in. I'm drawing a mess, but in this case, it's actually more, nicely compactified in here, but it is linear. It could be that in terms of this linear piece, we've got a linear piece that's very similar to this over here. We could cut and rejoin, and thus we might switch these two, and so this linear piece that used to be over here is now here. This linear pair now gets switched over here, and so you can also change your sequence via homologous recombination. There's many different ways to mute your sequence. Homologous recombination is one of them. For homologous recombination, all you need is for similar sequences. So you might have these two duplex DNA sequences right here, and maybe you get a nick. So maybe you got a bit too much sun and the UV light nicked your DNA. This does happen all the time. You might have something that actually needs repair, and so maybe you're having homologous recombination to repair something, but we're just showing the simple case right here, where this red strand now invades over. What you might have is that maybe these two sequences might be virtually identical. So yeah, you've got base pairing here, you've got base pairing here, but if these two sequences right here are virtually identical, well then instead you could have that the red base pairs to the blue and the blue base pairs here, and we can get branch migration, where now instead of the blue base pairing with the blue, we have the red and blue base pairing together. And so this red thing can insert itself in here, the blue insert in here, and it's perfectly happy doing that, because before we had blue and blue, but because it was basically the same sequence, we have the same sequence here, and so the DNA is just as happy. So if you have the same sequence or virtually identical, maybe there's a bit of a mismatch, maybe there's an A that is now needing to pair with C instead, but if they're mostly the same, you can get this, this recombination, this homologous recombination event happening, where one strand invades into the other. We create a holiday junction, branch migration, and now in order to finish the recombination, we're now going to have to resolve this this holiday junction right here. To see that a little bit better, let's take this bottom half and do a 180-degree rotation so that this now is over on this side. And so we can see blue, red, blue, red. This is the red strand down here, this is the red strand down here, and then if I do blue, red, that would be this blue and this red if I do a 180-degree rotation and move it over on this side. We can now see that there's two different ways to resolve. We can either cut horizontally. If we cut horizontally, we have this blue strand, the blue strand right here, and then we have blue, red, blue, blue, red, blue, and that would be your top new. So we would now have our new double-stranded DNA right here. We also have another one, the red here, but now it's red, blue, red, red, blue, red. So we also have our new double-stranded DNA. And so in this case, our homologous recombination went from this thing here to this right here. But remember, the sequences were virtually identical, possibly completely identical. And so this part right here is basically the same thing as this part right here. And so we don't really get recombinant products. We have basically the same thing as what we had before. Maybe there is one mismatch if the sequences weren't completely identical, but it's basically the same sequence that we had before. But instead, we could cut vertically. And so if we cut vertically right here, we now have blue, red, blue, red. So blue, red, blue, red. And on the other side, blue, red, blue, red, blue. So red, blue, red, blue. And so in this particular case, we do get recombinant products because this right here is quite different from here. What we've basically done is exchange the tails. So these two tails, they get exchanged right here. And so we now have homologous recombination creating a very different DNA sequence right here. And so that's what occurred in this one right here, is we now have homologous recombination occurring, and so there was some switching right here. So those are examples of some types of things that can happen to change your DNA sequence, and that change allows evolution to occur. And so we might be interested in a phylogenetic tree to figure out what happened in terms of the DNA sequences. How are the DNA sequences related? And we can see in this particular phylogenetic tree that animals and fungi, for example, are more related than, for example, cyanobacteria. So the DNA sequence between animals and fungi will be more closely related to cyanobacteria. To determine what's closer, one can look at distances between sequences. And so one common distance between sequence is a Hamming type sequence, where we first do an alignment between the sequences and then notice the number of differences between them. So here, if we take a look at one gene that's common between the cat, dog, rat, and cow, we can see that between the cat and the dog, the sequences are the same, except for this T changed to a C and this G changed to an A. And so we had 2 mutations within this 10. So we've got 10 bases right here, 2 mutations within 10. So we've got a 0.2, and so we say the distance here is 0.2. So if we compare the cat and the dog, it's 0.2. Between the cat and the rat, there would be 4 bases changed, and between the cat and the cow, there would be 7 bases. And so we can come up with this distance matrix. And notice between the cat and cat, it's 0 right here. So this is a standard distance matrix comparing 4 organisms, so we get a 4x4 matrix. There are many different types of distances that one can calculate. This Hamming distance would be one of the simplest. And there's lots of software that will calculate distances. So if we have a distance matrix, we can then use that distance matrix to say what organisms are close to each other. We can also take a look at particular proteins. So in this case, we're taking a look at the myosin superfamily of proteins. And we can either compare the amino acid sequence directly or go back and compare the DNA sequences that then coded for the amino acid sequences. And 2 proteins that are similar are going to be close on the tree. those that are further apart will be further apart on this tree. Let's take a look at a artificial example that I have borrowed down here. In this case, we get a perfect tree-like distance. The distance from the cat to the dog is 3, and we can see the distance 1 and 2 is 3 here. These distances might represent that Hamming type distance, they might represent evolutionary time, they can represent various different things, but we have some kind of distance. And we want to say that at some point a cat and a dog, they're similar to this, this point right here. Not the cat is more similar to the dog than it is to the rat. In this case, the distance is 4. If we take a look at that distance of 4, 1 plus 1 plus 2 is 4. Rat to cat, the distance is 5. Cat to cow, the distance is 6. Dog to cow, 7. Dog to rat, 6. So this is what's called a nice additive tree, where all these distances make perfect sense with respect to the tree. That doesn't normally happen, but it can happen, at least in artificially created examples. But suppose we take 2 different genes. So maybe one gene gives us this distance between dogs and cats and rats and cows, but a different gene gives us this distance. You'll note, to make my life simple, all I did is I changed my rat to a cat. So I had listed cat first and then rat. And so we have basically the same thing, but our rat and cat have changed locations on here. And that can happen. You can take 2 different genes from the same organism, compare them among multiple organisms, and come up with 2 different trees. So we have 2 different trees right here where our cat and rat have changed spots within the leaves of these trees. But we're supposed to have one phylogenetic tree that describes how cats and dogs evolved, from from ancestors. Well, in reality, trees may not be the best description of this evolutionary process. We may have, and also in this particular example, we had perfect trees for both of these. We had nice additive trees. We could instead have maybe a linear combination of these 2. So in this particular case, I took the average of here. So between the cat and the dog, it was 3. Between the cat and the dog, it's 5 in this tree. 3 plus 5 divided by 2 is 4, so that's the average. And so if you average these values, you can get to this thing here. Maybe it might be linear combinations that we might be interested in. And so maybe one thing we want to do is take our 2 trees that don't, that are not in agreement, and take a linear combination of our distance table, maybe with some weight for some biological reason. Maybe we want to weight this one more, or maybe we want to just take the average to get this thing here. And then the question is, well, now we no longer have a perfect tree. This may not, most distance matrices, if you just take an arbitrary distance matrix, it's not going to give you this perfect additive. They won't be additive. You won't get this perfect, tree right here coming from this distance matrix. And if we do this linear combination, from these artificially created things, it's unlikely to create a nice, perfect tree from it. So what should we do instead? Well, there's other things that one can have too, is instead of just this vertical over time, so we might have, mutations, in this organism, you might have 2 different mutations creating 2 different progeny, additional mutations creating more progeny, but you can also have things coming together in here. Remember when we took a look at, suppose we take a look at, maybe you are very unlucky and you get infected by 2 different HIV viruses. So you might get infected by 2 different HIV viruses. Well, let's draw an HIV virus right here. So maybe you've got your HIV virus 1 and your HIV virus 2. Well, if both of these are within your cell, you can get recombination between these. And so you might get recombination where you've got the red here and the red here, and the blue and the blue here. And so it could be that these 2 HIV viruses combine to create a new virus, and the DNA comes from both of them via this recombination event. Well, that's a good thing for us algebraic topologists, because we've just created homology here. We've just created homology. When these 2 things come together, we have homology in our tree. And so a tree may not be the best things. We can have these things called reticulation events. And so reticulation events, so when you listen to Rabinon's talk in the December workshop, he'll use the word reticulation event, and that's when you've got this melding of lineages that may come from a recombination event or from something else. And so maybe a tree-like structure is not the best model. Maybe we need something that has interesting first homology. Another thing that can happen, and this is quite common for the influenza virus, is you might have reassortment of your genes. So in the influenza virus, you basically have 8 DNA segments, or 8 chromosomes. So you've got 1 DNA segment, a second DNA segment. If you count them, there's 8 disjoint DNA segments. Well, you might get infected by 2 different influenza viruses. This does happen. And so you can get sick from 2 different ones, or so you might get sick from 2 different ones. They come into your cell. So this is your cell. The viruses have this protective coating right here. So you've got this nucleoprotein capsid that encapsulates your DNA. When it goes into your cell, that capsid goes away, and you're just the DNA now. This is viral in this case. We've been mentioning DNA, but these mutations can happen with RNA viruses as well. So we might have an RNA virus. So this could be DNA sequences, or it could be RNA sequences. The example we're looking at right now is RNA sequences, and so these are actually RNA sequences. But the RNA sequences, they get into your cell. Eventually your cell might die, and so it might want to then go ahead and reform encapsulate itself into this virus, so it can then infect other cells instead. What can happen though is there can be intermixing. So maybe you've got this L virus and you get the L virus out. You might have the M virus, you might get the M virus out, or you might get an intermixing between the two. And so most of the sequence is from the M virus, but you'll notice that there is one sequence from one of the DNA segments from the N, and so this is where you've got your reassortment. So it's not recombination. We haven't cut any RNA, but the RNA segments have been reassorted. We now have 7 blue segments and 1 pink segment. Well, we can go ahead and use persistent homology. In this case, we're taking a look at just one of the. So we had the 8. Let's see, do I have that picture? And the next one, we have the 8 ones, but in this particular case, we'll just take a look at one of these. So just taking a look at one. I don't remember which one we're looking at. If we take a look at just one of them and take a look at the 0th dimensional persistent homology, you can see that we have a lot of different subtypes for our influenza virus. But if we then take a look at as their distances, so we've got several different influenza viruses, but we can take a look at a Hamming type or another distance between them. As we grow our epsilon balls, they join together, and we can create a tree using our 0th dimensional homology. It's not the standard way, but it is one way of doing it, is that as these things come together and coalesce, so as when 2 of them get close enough to come to an edge, then we can join them together. And so when these 2 generators get killed by an edge, we'll join them together in the tree. And so if now this generator and this generator gets joined together, so we now have these 3 get joined together, then we can also join them together, and we can create a tree like that. In this particular case, there was no H1 and no H2. There was only H0, so we only had H0, and that's because we looked at only one of the RNA segments. We looked at only one RNA segment. And for influenza virus, there's basically, for influenza virus, there's no homologous recombination. And so the only sort of exchange you get is from that reassortment. So you don't get homologous recombination here. And you see that by taking a look at one segment. There's no intermixing between these things. But if we take a look at, so in this case, if you take a look at the HA, or if you take a look at the NA, we only have 0-dimensional homology. So there's no recombination between any of the HAs or between any of the, if we look at NA separately, there's no recombination between any of the NA genes. But between the NA segments. But if we take a look at NA combined with HA, well, now there are some of these things where maybe you'll get your, the NA may come from one of the viruses and your HA may come from the other virus. And so you do get this first homology where 2 of these things might come together. So we had these 2 viruses. So we had an initial virus, it mutated to create 2 new viruses, but then we had this reassortment to create this 4th virus right here. And so we can see this first homology where we had these descendants, but then these 2 descendants came together through a reticulation event, and this particular reticulation event was the reassortment. where DNA segments from these two come together and intermix right here. So that was this intermixing, so we might have created this R3 right here. Any questions up through here? So we can take a look at also higher dimensional homology. We can take a look at dimension 2 homology, and this right here is an example of dimension 2 homology. So these viruses right here, they are close. They're very close here, they're close here, and so we've got this closeness in terms of their distances, and so we can see some closeness in terms of this. So we do have non-trivial homology here, both H1 as well as H2. I want to take a closer look at this graph right here and what that graph means. What that graph means is basically when, in terms of this reassortment process, so you've got your virus here, and in your virus you've got these 8 different DNA segments. Some of these DNA segments, so 3 of the DNA segments, PB1, PB2, and PA, they all encode for RNA polymerase. That's the, so you've got your RNA that then gets into that cell there, and that RNA will need to be, a copy of that RNA will need to be made, and this RNA polymerase, these 3 different DNA segments, encode the genes necessary to create the proteins for to make copies of RNA. So this is for making copies of RNA. We can also have proteins that will encode for the nucleoproteins, so we can have this NA right here, HA as well, that sort of encode for these things here. You also have your matrix protein M1. You have a number of different proteins, RNAs that transcribe for these different proteins to create your RNA virus. Well, what this tells us is that when you go ahead and you mix the 2 viruses together to create this 3rd virus right here, what we have is that the RNA polymerase genes are going to likely come together. And so if I've got the red for my RNA polymerase, and I have another gene which is blue, well, it's unlikely for me to get red. Maybe that type of cell doesn't survive. So you don't get all different combinations. So if you came in here and got red, that's something that's actually unlikely to happen. And so this type of thing where we get a mixture between our segments coding for RNA polymerase, they are unlikely to, they're likely to segregate together. So you're not likely to get intermixtures, you're likely to get either all red or all blue. Blue was probably a bad choice. Here there's blue was things that, yeah, these are likely to be together, so most likely NA and HA will also go together into the same thing, but there's less of a, with this line thickness being less, it's less likely, so there's more likely of a chance that you might get, one copy of the NA from here and then a copy from HA from over here instead. So the type of segregation that you'll get in terms of creating these things here, what will create your new virus, So we can get that information by taking a look at your persistent homology of reassortment, what is likely to reassort and what is likely to maintain within the same virus. So we will finish up talking about viral evolution on Monday, as well as discuss a new topic, but more persistent homology applications on Monday. Any questions? See you Monday.